one gene determining a single character is called as polygenic inheritance. Example, skin color in man. Okay, just now we discussed skin color in man is an example for polygenic inheritance. Okay. Next we will quickly see about recombination. What is recombination, guys? Where does recombination happen? Chromosome, very good. DNA, okay. In which phase of cell division or in which stage of cell division does recombination occur? What is recombination? They will combine, the chromosomes will recombine, okay. They will overlap on each other and they will exchange their genetic material. Yes or no? That is what we call it as recombination. Yeah, when does recombination happen? First question. During anaphase. Anaphase by recombination. We did cell cycle and cell division. Where do you think? Yes, it happens in meiosis 1. It happens in prophase 1 of meiosis 1. Okay, these are very basic questions, you cannot forget this. Okay, so recombination happens in meiosis. When does meiosis happen? Where does meiosis happen? Let us put it like this. Where does meiosis happen? Gametes. Yes or no? Gametes or reproductive cells. Okay, so recombination will happen during gametogenesis. Okay, what is gametogenesis? It is a process of formation of gametes. Okay, so everything should be understood using logic. You simply can't by heart all this. Okay, fine. So it happens in meiosis. In meiosis there is a stage called prophase. In prophase it happens in Facetin stage. Okay, in prophase, Facetin, pairing of chromosome will happen. Okay, and in zygotine you will have in which stage does crossing over happen? Facetin or zygotine? Who will tell me correctly? Zygotine. Facetin pairing will happen. Zygotine you will have crossing over, basic concepts. Okay. So, Facetin you have pairing of chromosome. In zygotine you will have crossing over. Okay. So, you will have the homologous chromosome. Homologous chromosome means similar chromosomes. Okay. Listen to me carefully now everyone, any doubts? Okay. So, homologous chromosome will first pair with each other. Okay. The pairing of homologous chromosome is called synapsis. Okay. What is the pairing of homologous chromosome called? Synapsis. Okay. And this synapsis happens with the help of a complex called synaptonemal complex. Okay. It is a complex protein which is helpful for pairing of homologous chromosome. Okay. So, first you will have synapsis happening, then in the zygotin stage you will have crossing over. So, what is crossing over? Exchange of genetic material like this. Okay. Fine. So, this crossing over, the point of crossing over is called, who will tell me? What is the point of crossing over called? Very good, chiasma. Okay, this point of crossing over is called chiasma. Okay. So, now you will have the genetic material being exchanged. Just to show you the difference I am shading. Okay. You can see these are the point of exchange. Okay. And this is why we are all not the same. Crossing over is like for very important for variation among organisms. Okay, so you can have very important questions. Where does crossing over happen? During gametogenesis. Okay, during which stage does crossing over happen? During meiosis 1, prophase 1, under that zygote. Okay, under the zygote. And but where does pairing of homologous chromosome takes place? Phacetin. What is the process of pairing of homologous chromosome called? Synapsis. Okay, and these 4 chromosomes, the paired chromosome are called tetrad or bivalent. Don't ever forget this. Okay, definitely one question will be asked based on this crossing over and things. Okay. Yeah, and this crossing over is responsible for variation among organisms. That is why you are not exactly similar to your parents, nor you are exactly similar to your siblings, except for identical twins. All of us are different. Okay, fine. So next we will see a small topic called linkage. Okay. What is linkage? Can anyone tell me? Physical association of genes. Okay, then the genes will be they will be close to each other. Okay, say for example, this is the chromosome. Okay, you will have 2 genes A and B which will be close to each other. Are you guys understanding? And you will have one more gene C and D which are far from each other. Are you guys understanding? The genes which are close to each other are called linked genes, and these linked genes will be inherited together. Yes or no? Yes or no? The linked genes will be inherited together, whereas the unlinked gene will not be inherited together. Okay? Linkage is the genes which are close to each other and they will be inherited together. Okay, so or alleles, we can't call it as genes, they are alleles. Okay, alleles will be inherited together and they will also be close to each other. When will they be inherited together? When they are close to each other. When they are farther, is it possible for them to get inherited together? No. Okay, so you have 2 kinds of linkage, complete linkage and incomplete linkage. Okay, complete linkage means the genes will stay together after inheritance. Okay, in incomplete linkage they will not stay together. Very simple. Okay, so here if they are going to produce gametes, it will be, they will again still stay together. But whereas this, there are chances that they will get separated. Are you guys understanding? So this distance is measured in terms of recombination frequency. The distance between 2 genes is measured in terms of recombination frequency. Okay. Higher the recombination frequency, greater is the distance between them. Greater the distance between them, lesser will be the linkage. Okay. Are you guys understanding? I will repeat. If the recombination frequency means what to say, the distance between 2 genes are measured in terms of recombination frequency. Okay, greater the distance or greater the recombination frequency, there are very less chances that they will be inherited together. Yes or no? Yes or no? When the genes are far from each other, the chance for them to get inherited together is less. In that case, you will have incomplete linkage. Okay, so lesser the recombination frequency, okay, greater the chance of linkage or complete linkage. Okay, greater the recombination frequency, there is a very less chance that the genes will be linked with each other. Okay, so linked genes, don't you think it's something against independent assortment? What does independent assortment say? 2 genes will independently assort. But what is linkage telling? The 2 genes will be inherited together. Don't you think there is a deviation? Are you guys understanding what is independent assortment telling? Whenever there are 2 genes, they will segregate independently is what your independent assortment telling. But what is linkage telling? The genes which are closer together will be inherited together. Okay. So Mendel had some loopholes in his work. Okay, Mendel had some loopholes in his work which was later studied by many other scientists, and they started proposing multiple allelism, codominance, linkage, pleiotropy, polygenic inheritance. These are all non-Mendelian inheritance. Okay, they do not hold good for Mendel's law. Okay, they do not what to say, they do not stay true for Mendel's law. Okay. So, linkage is something against or something that deviates from Mendel's law. Okay, fine. So, who discovered linkage? Anyone? Any idea? Which scientist discovered linkage? T.H. Morgan. Okay, T.H. Morgan was the one who discovered linkage. He used Drosophila melanogaster. He used Drosophila melanogaster for his experiments. Okay. What is parental type and what is recombinant type? Anyone has any idea? Shall I erase this? Everyone understood? Okay. Say for example, I will take a simple cross AA and AA. So, the F1 will be A, right? Again, what will be the F2? So, in this, which are the parental type? These are parental type. They exactly look like correct? One second, sorry. A. Okay, yeah. Which are the parental types which exactly looks like your parent? So, you have this and this are your parental type, whereas this is a recombinant type. Are you guys understanding what is parental and recombinant? Parental means they have the exact genotype as that of the parent. Recombinant is something new. That is it. Okay, fine. Next we will move on to sex determination. Okay. So, what is this chromosomal theory of inheritance? Just now we did know recombination, crossing over. Okay. So, it is all based on the chromosomes. Okay. So, as I already told you, 50% of our chromosomes we get from our father and the other 50% we get from our mother, and all these genes come together to form all the alleles come together to form a character or to determine a character. Okay. So, now let us move on to sex determination in organisms. Any doubt in this? Still here? Still here, any doubts? Okay, write down. So, first you have the first type of sex determination is XX and XY type. Okay. Here the female is a homogametic sex. What is homogametic? Means they produce similar gametes. Okay. So, when they produce, all the gametes will contain X and X. Yes or no? Yes or no? Once they segregate, there all the gametes will contain only X chromosome. So, female is a homogametic sex. So, you should understand this word homogametic. Homogametic means all the gametes are same. Okay. Next, here it is XY is a male, it is a heterogametic sex. So, half of the gametes will contain X chromosome and the other half will contain Y chromosome. Okay. So, which gender determines the sex? Which gender determines the sex? Male. Okay, that is very important. So, here male is the one who is going to determine the sex. Okay. the gametes of the male is going to determine the sex. That is what we call it as XX, XY type of chromosome. It is also called as male heterogamety. Okay. Next, what is the next type? What is the next type, guys? No, you have XXXO type, right? One more. Can anyone give me your elite book? What is this XXXO type? Open the lesson. What is XXXO type? Here there is no second chromosome. This O stands for 0. Okay, there is no second chromosome here. It's only one X chromosome. Okay. Yeah. So here the XX is the female, okay, and XO is the male. Okay. So what will the female produce? The female will produce XX, the male will produce only X, and here there will be no sex chromosome in this gamete. Okay. So again, who will determine the sex? The male. Okay. Next, ZW type. Okay. Here ZW is female, very important. Okay. So here female is heterogametic. Are you guys following me? And here the male is homogametic. So who is determining the sex here? Female. So the female will produce Z and W. Here the female will produce Z and Z. So here you have your female to be the heterogametic sex. Okay. Then you have ZO method. Okay. Here the ZO is the female. Here it is male. Again here the female is heterogametic. Male is homogametic. It's just the other way around. Okay. If you understand XX, XY, in both the cases, who is homogametic? Female. In this both the cases, who is homogametic? Male. Okay. So example for XX, XY method. Examples are important, guys. Mammals. Okay. Examples for XO method, you have roundworms. Example for ZW type, it is birds and reptiles. Okay, and example for ZO method, it is moth and butterfly. Any doubts? Done? Everyone understood sex determination? Next, write down the heading mutations. Can anyone define me what is mutation? It's a sudden, heritable or inheritable change in the gene. Okay. Very, very important. Mutations are sudden, heritable. When do I call something to be a mutation? When it can be inherited to its offsprings. Okay. Heritable change. It can be of three types: genomic mutation, okay, chromosomal mutation, and you also have your gene mutation. Okay. So genomic mutation means, what is genome? What is a genome? The total DNA content of the cell is called genome. Okay. So if the entire genome, like increasing the chromosome level, change in the chromosome number. Okay, change in the chromosome number is called genomic mutation. This is commonly seen in plants, polyploidy. Okay, 2n will become 4n, 4n will become 5n, like that. That is seen in plants. Okay, then you have chromosomal mutation. It will just change the deletion or addition of chromosome. Have you heard about chromosomal aberration, Klinefelter syndrome, one extra X chromosome, Down syndrome, one extra 21st chromosome, Turner syndrome, one less X chromosome? Yes or no? It is because of deletion and addition of chromosomes or a part of a chromosome, or its parts, of chromosome or its part. What is gene mutation? It is a mutation on the gene itself, like sickle cell anemia. Okay, sickle cell anemia is an example for gene mutation. Only that particular gene, in that gene only one nucleotide was changed. Yes or no? GAG to GUG, right? A was changed to U. Okay, so that is what we call it as gene mutation. A mutation at the gene level is called as gene mutation. Sickle cell anemia, your hemophilia, they are all examples for gene mutations. Okay, yes. And other comparison is you have somatic mutations and germinal mutation. Can anyone tell me what is somatic and germinal mutation? Somatic mutation happens in the somatic cells, germinal mutation happens in the reproductive cells, that is in your testes and ovaries. Okay. So you have in genomic mutation, you have euploidy. Where is genomic mutation? Okay. You have euploidy, polyploidy and aneuploidy. Okay, genomic mutation is of 3 types: euploidy, polyploidy and aneuploidy. Okay, euploidy means 2n becoming 3n. Are you guys understanding? 2n becoming 3n is called euploidy, or an becoming 2n. One set of chromosome is increasing. That is what we call it as euploidy. Okay, in polyploid more than one, like tetraploids. Okay, you have your 4n. This is very commonly seen in plants. Tetraploidy is commonly seen in plants. Okay, then you have pentaploid. Pentaploid is 5n. Like this, it just simply goes on. Okay, and then you have aneuploidy. Aneuploidy is change in addition or deletion of one chromosome is aneuploidy. Okay, it will change the chromosome number. It will either increase the chromosome number or either it will decrease the chromosome number. Okay, so aneuploidy is of 2 types. One is trisomy and monosomy. Very good. Okay, trisomy is addition of chromosome, monosomy is deletion of chromosome. Okay, so one best example for trisomy is Down's syndrome. Yes or no? Down syndrome has which chromosome extra? 21st chromosome, 21st trisomy. We also call Down syndrome as 21st trisomy. What are the symptoms of Down syndrome? Flat forehead, epicanthic folds, very small nose, right? Mental retardation. The person will not be very alert. They are all the symptoms, or not symptoms, they are all the characteristics of Down syndrome. Okay, the person will be born with these characters. This is because of a genetic problem. Are you guys understanding? Then Klinefelter syndrome. Klinefelter syndrome, which trisomy is it? X. You will have XXY. Okay, so here Down syndrome, how do you write it as? Usually 45 autosome plus XX. Are you guys understanding what I have written here? We all have how many chromosomes? 46, 23 pairs, right? So 44 plus XX or 44 plus XY. So I have written it as 45 plus XX or 45A plus XY. Okay. Then what about Klinefelter syndrome? 44A plus XXY. Are you guys understanding? 44A, they can give you this and they can ask you to find out which disease it belongs to in your options. Okay. So this is Klinefelter syndrome. So what is the symptoms of Klinefelter syndrome? Female-like characteristics. Okay, is the symptoms of or it's a characteristic. I can't call it as symptom since it's a genetic disorder. I can't call it as a symptom. We can call it as the characteristics. Okay. Then you have Turner syndrome. Turner syndrome is 44A plus XO. Here there is no one extra X chromosome. That is what we call it as Turner syndrome. What is the characteristics of Turner syndrome? Short stature, webbed neck, okay, underdeveloped ovaries, very poor reproductive system, the metabolism will be very slow. They are all the symptoms of Turner syndrome. Okay. And when is the chance of all these genetic disorder will increase? When the mother has crossed the age of 35 or 30. Okay, if the mother is planning for a pregnancy after the age of 30 to 35, the egg deposits would have reduced by that time. Okay, so there will be a lot of, the egg will not be very healthy and as a result you can get this, and there will be a non-disjunction during separation. So you all know during anaphase the chromosomes will separate, yes or no? Everyone remember that during anaphase the chromosome will separate. So because of the poor quality of the egg, sometimes extra chromosomes will be pulled towards one cell. Are you guys understanding? Or one chromosome will be left out. Okay. So this entire aneuploidy is caused because of non-disjunction during meiosis. So when I talk about non-disjunction, it is because of improper separation during meiosis. Did you all follow me what we are trying to understand? Okay, fine. Yeah, pretty much we are done. Okay. Now let us quickly understand the gene mutations. Okay. Now we finished genomic mutation. Now we will go to gene mutations. Okay. Shall I erase, guys? Did everyone understood what is trisomy, monosomy and all? Any doubts? Any doubts? Okay. Now we are talking about the genetic level, we are talking at the nucleotide level, okay. So addition, deletion, all that we are going to talk about, okay. So first I will just write a gene sequence. Okay, I have just randomly written. Okay. So now the first one is insertion, the first kind of gene mutation is insertion. What is insertion? I can insert a new nucleotide here. Okay, I can insert a new nucleotide here. What will happen if I insert a new nucleotide? Will it change something? It will change the sequence of codes. Okay, so if protein will change, how the protein will change? The amino acid will change. Okay, so initially AAT will be read as one codon and AAT will code for its corresponding amino acid. But now when I am introducing this AAA, yes or no? Are you guys understanding? The amino acid will be totally different. Okay, so addition of a gene, addition of a nucleotide, not a gene, addition of a nucleotide can change the coding sequence. Okay, and this kind is called as the frame shift mutation. It is going to shift the frame. okay, or leave this, okay. I am adding somewhere here, I am adding U